Good morning, everyone. Good afternoon, good evening, wherever you are in the world at this time. I welcome you all to this inaugural session of our monthly live series, BISMiS Live, which we are going to start today. Our speaker today is Professor Philip Hugenholtz. He's the director of the Australian Centre for Ecogenomics from the University of Queensland. His work has revolutionized our understanding of microbial ecology evolution over the past 26 years of his research career. He did his PhD back in 1994 from University of Queensland, and then he moved to US and he joined the Joint Genome Institute, where he worked until 2010, where he worked on setting up microbial genomics, especially metagenomics. And then late 2010, he moved back to Australia to set up the Australian Centre for Ecogenomics. But his career is highlighted with a lot of fellowships and awards that he has received. Very recently, he's been awarded the Australian Research Council Laureate Fellowship for delivering 100,000 genomes of uncultivated microorganisms that represent the microbial dark matter. He's published over 400 papers in various formats, and 39 of them include science and nature publications. He's also been awarded the Young Investigator Award by the International Society for Microbial Ecology. And in 2012, he was awarded the American Academy of Microbiology Fellowship. And in 2017, he was elected as the Fellow of the Australian Academy of Sciences. That has been, an illustrious career, and we hope that by joining for this inaugural talk, everyone will be benefited by what Phil has to share and the lessons learned on the genome taxonomy database. So Phil, welcome, and thank you for accepting our invitation. Thank you.
Well, thanks very much for that kind introduction, and also thanks for the invitation to speak at this inaugural series. And I'm very impressed to see so many people tuning in. It's a Saturday afternoon here in Australia, and I think it might be really early morning on a weekend where some of the folks are. So I do appreciate everybody's time. So I'm going to talk about the Genome Taxonomy Database today, or GTDB for short. I'm going to give, I've given an introduction to this database on and off now for since 2017. So some of you may have seen me give that introduction or give that description of it, but I'll do it again because I can't assume that everybody knows about it. That'd be nice if they did. And this is actually supported by the ARC laureate that was mentioned before. And you can see now actually we're well above that 100,000 genomes, heading towards 200,000 here. So let me begin. I have to click on it to get it to go. So I would say it's fair to say that taxonomy has a pretty poor reputation. I like to show these bits of feedback I've received over the years. One was a tweet with the pronouncement that taxonomy is dead. I think quite a few people would like to see it dead. Just let it die in peace. And then this was written on a tissue at a presentation I gave a couple of years back, presumably in response to my presentation. So taxonomy, what is it good for? Zilch. But I would say, from my observation of it over the years, there's a pervading sense of frustration amongst researchers because nobody particularly likes taxonomy except for a few enthusiasts, but everybody effectively has to use it at some point if they're doing microbial ecology or evolution. So I think we can start from the basis that taxonomy is a useful human-made construct. If you don't believe that, you might as well tune out now because the rest of the talk is based on that presumption. And I'd say it's fairly widely believed that taxonomy should ideally be consistent with evolutionary relationships or the phylogeny of organisms, although of course it can be based on anything. And it has been based on other things in the past, such as phenotypic properties, which don't necessarily reflect evolutionary relationships. Microbial taxonomy is currently based on 16S RNA relationships. I feel we are beginning to have a transition at the moment away from that, but I would still say that the 16S RNA gene is a very important taxonomic marker that's been used very extensively over the last quarter of a century. So the problem is that microbial taxonomy does not directly translate to phylogeny currently. And this is due to a few things. One is historical artifacts. I mentioned the phenotypic classification that don't necessarily match phylogeny. So case in point being organisms that might produce spores, rod-shaped and gram-positive would maybe get classified as Clostridium, but in fact represent a much broader diversity of organisms. And I'll bring that up again as an example later on. Then there are limitations with ribosomal RNA gene trees, 16S ribosomal RNA gene trees, even though they've been a spectacularly successful objective marker for inferring evolutionary relationships between organisms. First of all, they have limits, resolution limits at both ends of the spectrum. So because they're so conserved, you often do not get species level resolution, and particularly if you're basing your analysis just on a fragment of the 16S molecule, which is very common these days. And then at the other end of the spectrum, it's often hard to get a high resolution or good resolution of deeper relationships. That may be impossible generally, but often it's down to the actual gene. And I guess you're asking a lot. You're asking 1,500 nucleotides to faithfully give you interpretation of 4 billion years of evolution. So that is quite an ask. We have historically suffered from inadequate reference sequences of isolated organisms, and I'll get onto this in a lot more detail later on in the talk. And then you do have some insidious artifacts with 16S RNA. If you're using PCR, you get these chimeric organisms forming or sequences forming, which are indicating the presence of organisms that don't actually exist, and that just muddies the water in general. So I would say now trees that are based on more of the genome, I've written genome trees here, but technically it's not the whole genome, but often not the whole genome, but basically phylogeny that's based on a larger amount of the data that's coming out of a genome appear to be the most promising basis for a microbial taxonomy today. And this has been made possible because we're now able to generate genomes rather trivially, as opposed to, say, when I was doing my PhD, it was a huge undertaking in fact, it hadn't been done for a bacterium to sequence the genome. And when I was at doing my postdoc, for instance, it was like that was a whole PhD for somebody to sequence and analyze one bacterial genome. These days, they're just generated very rapidly for relatively cheap. And that goes for both cultured and uncultured organisms. So to give you an idea, this is what's been going on. Genomics and metagenomics have been evolving on the back of high throughput sequencing. So this was a study back in 2004 of acid mine drainage ecosystem for metagenomics with Jill Banfield's group. And 76 million base pairs of Sanger sequence data were generated at a fairly high cost of, I think it was somewhere in the order of a few $100,000 US. Then when so-called next generation sequencing came in, such as Illumina, the cost dropped right down and the amount of throughput went right up. So this is a Kauerman study at JGI in 5 years later, 2009, with 17,000 million bases, or 17 gigabases. The following year, a Chinese European consortium, MetaHit, produced 570 gigabases of Illumina data. I can still remember how amazing that seemed, that anybody could generate that amount of data, and what could anybody possibly do with it. And now today, at the latest incarnation of the Illumina instrument, the NovaSeq, produces 3 terabases of data on a single S4 chip. So really mind-boggling improvements. And this is what's driven the whole genomics field. And by the way, I'm sure many of you know that this is not the end of the story for sequencing, because now we have long-read sequencing like Nanopore and PacBio, which may produce another major revolution in genomics. So in short, in the 2000s, because of the amount of data and the expense of the data, you could generate millions of base pairs of sequence data. And with that, there's just not enough data to recompile the genomes of organisms from that ecosystem. And so the best you could do with those data at the time was to annotate individual reads and then make gene-centric, so-called gene-centric profiles in order to learn something about the community. Now, as of the 2010s, with this increased throughput, sequencing throughput, we can generate gigabases of data, and that allows us to basically reassemble component populations within a community to produce these so-called metagenome assembled genomes, or MAGs for short. So you've probably all heard about MAGs. It's definitely taking over in the genomics space. And once you have a MAG, or a population genome, you can do all of the usual things that you do with a genome from an isolated microorganism. You can do a metabolic reconstruction, you can infer what the organism might be capable of, and you can produce a genome tree on which to base a taxonomy. So there's been a huge data deluge in the last few years, courtesy of these MAGs. So here's a study from 2018 where 2,631 MAGs were assembled in a single study from an oceanic source, and everybody's blown away. And then around the same time, here at the Australian Centre of Ecogenomics, we mined the sequence read archive and pulled out almost 8,000 genomes. We thought that's an amazing thing, that we were all amazed by that. And then very shortly after that, Nicholas Capiti's group at the JGI pulled out 60,000 MAGs in one study. So starting to get to be, mind-boggling numbers in individual studies. And that was superseded from Rob Finn's group shortly thereafter with 90,000, and finally from Nicholas Segala's group and in the same year, more than 150,000 MAGs per study. So this is just mind-boggling. It is to me, having lived through genomics in the early days. But it's really the great positive side of this is it's fantastic, because what it's doing is it's giving us access to all of these organisms that can't be grown. Now, I haven't talked about that, but the great majority of microorganisms have not been obtained in pure culture. So we've been blind to the majority of microbial diversity since we discovered them. And so now we really have the technological capability to fill in all those gaps very rapidly. So data at this scale present a lot of, is in addition to opportunities, a lot of technical hurdles for analysis. One of them is what we're interested in, which is the classification or taxonomy. And I would say with this deluge of MAGs, we're taxonomically unprepared. So this is the deluge, and here's our current taxonomic practices. And we need to adapt to this in order to scale with this. And so that's where the idea for the genome taxonomy database came from. So the objective was to create a genome taxonomy that would objectively classify MAGs and that scales with the volume of data, and at the same time fix the current highly flawed taxonomy that we had available. So some of the things we were looking for is that the taxonomy should first and foremost be based on a sound phylogenetic framework, so a good tree. It should resolve polyphyletic groups, so groups that are appearing in multiple places across the tree should be divided and made into monophyletic groups. And it should take relative evolutionary divergence into account. So that last one is less familiar to people. And this is an example of why we feel that relative evolutionary divergence matters. So this is a paper, it was published in 2014 by Yauser et al. And it was a paper based on 16S sequences suggesting thresholds for defining different taxonomic ranks. And you'll see that, and this is based on the Silver database and their curated living tree project. And so a very reasonable idea. And the threshold that they came up with for defining a phylum was 75%. Now, the issue with that is that if you're just using sequence identity, you're not taking relative evolutionary divergence into account. That means that different organisms are evolving at different rates. So if you just use a flat sequence threshold, you're not really comparing apples with apples. And the example that I most commonly give is of a fast evolving group such as Mycoplasma. So if you use the 75% threshold, that genus constitutes 2 phyla. So, and we know that because it's a host-associated organism, it's estimated to have diverged from sister taxa around 400 million years ago. And the best guess, or the best estimate of the primary diversification of phyla, for which this threshold would suggest Mycoplasma represents 2, is 2 to 3 billion years ago. So you can see there's a disconnect. And so what's happening is that the Mycoplasma is evolving at a fast rate. And so using a threshold like that will give a misestimate of how ancient that lineage actually is. So just a few quick slides on the database itself. So we were looking to pull the information out of the INSDC databases. So we used RefSeq and GenBank. We initially complemented it with some mags that we'd mined ourselves that weren't currently in the database, but we now completely get all of our sequence data from the INSDC databases at GenBank and RefSeq. And they actually do releases every couple of months, which we cannot keep up with. So we decided to do every 3rd to 4th release, depending on how we were going. So the first thing we do is we estimate the quality of those genomes that we're pulling down from the public repository using Check'em, which is a piece of software developed here at the Australian Centre for Ecogenomics, and then call the genes. We complement that with type material. Now, I'm going to be talking about type material a fair bit in this talk later on, but it's really important because they effectively are your signposts for your taxonomy. They're the described organisms, and this is the type species or the type strain, and that gives us a taxonomic signpost that we can then use for annotating the tree. And so that information was pulled from a number of different sources initially, NCBI, LPSN, DSMZ, and Strain Info. Strain Info no longer exists. LPSN is now under the auspices of DSMZ, and so it's been very well maintained as an important resource. Then once we had done that, we would then pull out conserved markers for making the tree, and so out of the subset of sequences that pass the quality assurance step or the quality control step. And so just to give you an idea, in release 86 of Gtdb, there are about 100,000 RefSeq genomes, 24,000 GenBank genomes, and a supplement of 3,000. Okay, so when you have that many genomes and you want to make a tree, it's a good idea to dereplicate, because you don't want to make a tree that's got 8,000 E. coli genomes in it or 12,000 Staphylococcus aureus genomes. So we dereplicated using average nucleotide identities, and that allowed us to take representatives of larger clusters to make a tree that was smaller. The tree's still very large. In 86, it was almost 30,000 sequences, but sufficient that you can infer a genome tree. And we didn't want to use neighbor joining because that's a very crude, evolutionary distance is a pretty crude way of inferring a tree. So we wanted to use a better methods such as maximum likelihood, and there's a couple of tools that will scale. Well, actually one, FastTree, which was produced in the US, and you can actually make a tree of that size. And so we took 120 markers, as I mentioned before, conserved single copy markers, and we aligned them, and that produces a data set of about 30,000 amino acids. And so we had a matrix of 30,000 amino acids by 30,000 sequences and created a tree. And then after we had our reference tree, we would then decorate it. And so we decorated it with taxonomy that we pulled out of NCBI, and also sort of legacy 16S based information from Green Genes, which is now defunct, and Silver, so that we had a starting taxonomy on that genome tree. So once you have your annotated or decorated reference tree, there are two main tasks. The first one is to get rid of those polyphyletic groups I mentioned. So the poster child for polyphyletic groups is Clostridium, also things like Eubacterium, Bacillus. These are highly polyphyletic groups, as I'm sure you all know. And then the secondary task of normalizing the taxonomic ranks, and with Mycoplasma being the example that I gave. So for resolving polyphyletic genera such as Clostridia, you first of all identify the position of the type species, and there it is down there. And then everything else that's called Clostridium is not actually associated with that genus. And so these are polyphyletic groups. And so what we did in the first instance was just to suffix them so that we'd identify that they weren't actually part of that genus, but we retained the initial name because some of them actually have very well-known species of Clostridium. And if we change the genus name straight away, that could cause issues. But anyway, that's how we did it. And so in the initial paper that was published in 2018, we divided the genus Clostridium into 121 separate genera that span 29 families. You can do this at a higher level too. So these are, you're looking here at orders within the gamma Proteobacteria, the class gamma Proteobacteria, and the polyphyletic orders are shown by similar colors. So the most polyphyletic group here is the Ultramonadales, which split into 4 different groups, but also Pseudomonadales and these other groups are polyphyletic. And so you identify the group that contains the type, genus, and type species. So in this case, for the Ultramonadales, it's Ultramonas, it's here. And then the other groups, we renamed after the oldest validly named species within the group. So in this case, it was Schuanella, in this case, Psychomonas. This actually caused problems, which I will touch on as part of the lessons learned. I'll probably go into it in more detail then. So this second component, standardizing rank. So Donovan Parks, who's the lead developer on Gtdb, wrote a program called PhyloRank. And what it does is it uses the evolutionary divergence, which is in your maximum likelihood tree. So the maximum likelihood tree is estimating the rate of evolution of the different branches. So it's already doing that for you. And we use that as the primary criterion for circumscribing the taxonomic ranks by accounting for varying rates. And so this is the inferred evolutionary distances here. So this is what the algorithm's telling us, 1, 2, 1, 1, 1. And then what we do is that we set the root to 0 and all extent taxa to 1. And then we do a linear interpolation, which is a very simple interpolation from 0 to 1 across the whole tree. And what you can see is that in this case, this is a fast evolving group. And at this point, at this interior node between the root and the tips, this is 0.75 of the way down from 0 to 1. So that's the linear interpolation. And this much slower evolving group is also around the same area. It's also 0.71 down its trajectory. So these two groups here, even though they've got very different branch lengths and would have very different thresholds, just based straight up sequence thresholds, have similar evolutionary divergence to each other. And so based on the tree, you would predict that the ancestors that lived here at these interior nodes occupied the planet at the same time. And that's one of the things we want to do if we're interested in evolution, is compare ancestral organisms that coexisted if we're using ranks as that metric. So this is conceptually similar to a method called Trio2U by Donying Wu and Jonathan Eisen. And that's kind of where I took the idea from. They were using it for a different purpose, but that suited this very well. So as I said, linear interpolation is a very simplified sort of way to do it, but there've been, we and other groups have sort of been kicking this relative evolutionary divergence, or RED for short, metric, and it does seem to be pretty good if you compare it to more sophisticated methods, which I haven't got time to go into. So here's a visualization of when you apply this relative evolutionary divergence to a tree, and what you can see, it's like a contour map. When you're looking at an orienteering map or something, and you can see that the different ranks are projected on the tree, and you can see it contours according to the relative evolutionary divergence of this tree. And so everything that's, for instance, in this reddish group are what we would say are family level groups, and the explicit prediction is that they would have coexisted approximately at the same time on the planet. Now, so that's using, this figure is using the actual distances that the maximum likelihood tree puts out. But if you replace those with the red values, the 0 to 1 values, as you would expect, you get completely concentric circles. So that's how it looks. So this is associated tool. This part of the PhyloRank tool, I should say, is a way of projecting that information onto a plot. And so on the y-axis, you have the different ranks from species down to phylum. And on the x-axis, you have the relative evolutionary divergence or relative distance from 0, which is the root, to 1, which is the tips. And then each of the dots in this plot represents an interior node in your it's a decorated interior node in your tree. So as expected, we see that deeper groups such as phylum are closer to the root, and shallower ranks such as species are closer to the tips of the tree. But you can see it's kind of choppy, right? So it's not, you'd like to see it nicely, tightly banded like this way. I also just point out that polyphyletic taxa are shown in this plot as red, but monophyletic groups are blue. So you see outliers. So for instance, you'll see the family Nitrospiraceae. It's very deep for a family, and that's because it's just an underclassified phylum. And so people have not really broken that up into, it actually comprises many, many different families, but here everything's just lumped here, that there's a single family, a single order, a single class, and a single phylum. So it's underclassified. And if it would be more at home in a phylum level distribution, for instance. By the way, I forgot to say, these are like the goalposts. So the black line is the midpoint of the distribution, and then the yellow and red are just plus or minus 0.1. So you can see where the goalposts are, what we might be trying to go for. Then we have the beta proteobacteria, which is an example I often give, which is pretty shallow for a class and really looks more like it should be in order. So what we did is we just went and nipped and tucked all of those groups that were outliers to put them into a more orderly fashion, so that we can then say all of these interior nodes have similar range of red values. And so we're saying that all these classes coexisted on the planet at the same time. And it looks nice and neat, and it's appealing. And then you can see what we did, how we fixed up the problem. So that family level group was broken down into a phylum level, and that there was subsequent breakup of that phylum into lower level ranks. So that particular node is now actually a phylum level node. And then the order the class beta proteobacteria was made into an order within the gamma proteobacteria. Now this, we were trying to implement this more systematic classification and not cause too many waves, but in fact, it's pretty hard to do that. And so the Silver database has taken some of our suggested changes, and that was one of the ones they picked up, and this was picked up on Twitter. And they said, what's happening with these conflicting, what's going on? How is order beta a class gamma? And so what I would say, even if you looked at the earliest tree, which is in Berge's manual from 2004, even that tree already told you that the beta proteobacteria was more at home as an order within the gamma. That's the gamma proteobacteria. The rest of these are orders within the gamma proteobacteria. And you've got to remember that whole naming system, the alpha, beta, gamma, delta, epsilon, it was more or less an ad hoc naming system at the time based on a very small subset of the sequences that we now know today. So from a purely objective viewpoint, this is not a controversial move, but from I'm used to seeing the class beta proteobacteria, this is a very radical move, which has met with resistance. Okay, so this rank normalization process then, depending on which part of the tree you are, as you're adjusting everything by the contouring, either ended in splitting. So for instance, the order Clostridiales in the NCBI was subdivided into a number of new orders because it was too deep to be an order. Conversely, we saw some lumping as well. So for instance, the several classes that were class level lineages within the Bacteroidetes were too shallow, and so they were collapsed into a single class, Bacteroidea. So this is the kind of thing we saw. And then these other lines that are going around are indicating misclassified sequences at NCBI, or that's our opinion anyway. So this is a figure from the first paper for Gtdb, which is from 2018, Parks et al. So just a high level summary, and I probably already talked too much. This was just supposed to be the introduction part. There's 58% of the genomes for that release had one or more changes to the NCBI taxonomy. And this is looking at it across all the different ranks. And so that sounds like a lot, but if you look at it, mostly we managed to implement the new taxonomy, and the majority of taxa in the different ranks was unchanged. That's the green part. And then a smaller subset had active changes. And then this little gray or bluey gray thing, passive changes, those are actually gaps within the NCBI taxonomy where there's no information, it's just left blank. And so we filled those in. So they're called passive changes, whereas active changes were where there was an actual, had to change the name, like in the case of the beta Proteobacteria. This is an important one for all the nomenclaturists out there, that only 18% of the taxa in that genome tree, which includes quite a number of uncultured taxa, actually have valid names according to the prokaryotic code. And then a number have been proposed. These are could be, effective publication. I'll talk about that shortly. And then just note that the phylum has no valid names because phylum does not currently sit under the prokaryotic code, but it soon will. And then the majority of the names are actually placeholder names. So these are just names that are alphanumeric placeholder names like TM7, OP11, those kind of names. I've done a lot myself in the 16S world. So they're not Latin names, but they definitely don't fall underneath the code. So this is a screenshot of the website for the Genome Taxonomy database with the latest release. That is a picture of a giant tree down on this campus that I'm currently standing on, representing the tree of life. And it's there's a release every 6 to 8 months, depending on our workload. And the next release, which is 06 RS202, is imminent. So the naming system is, the first number is the GTB release, and then this is the RefSeq release. It's index 2. So remember I said we take every 3 to 4 RefSeq releases. So the next one will be RS202, 06 RS202. So as of release 04 RS89, we do a complete domain to species taxonomy, but before that, we weren't giving species identification to all taxa because it was just too much work for curation, for manual curators off a tree. And so we introduced an average nucleotide identity or ANI based species clustering in order to do that part, automatically do that part, and using type material as focal points around which we then created these ANI radii that we could then create these species, which is also used for the dereplication step. And so that means that we now, as of release 89, we have a complete domain to species taxonomy in the GTDB. There are a few tricky cases. Escherichia coli. Escherichia is a very notable one here, and we've copped some flack over the fact that we ended up trying to use Shigella, which is a known issue from a phylogenetic point of view, that it should not really exist. And so there are a few difficult cases like this that we'd had to work on. So we've had, over the couple of years that it's been live, we've had quite a few requests for different functionality. People like the idea of coming to a website and having access to a whole bunch of genomes in one place. And they're saying, oh, could you put in metabolic reconstruction, or could you make this and that and the other data available? But we really are very simply a taxonomy resource. And so we've kept it very focused on that. And this is the main interface page that you'll find when you go to the website, that you'll have the NCBI taxonomy here. There's an unfiltered version because there are a lot of ranks that fall outside of the canonical ranks that we use at GTDB, which is the phylum, class, order, family, genus, and species. We don't use sub or super taxa at all. And you can see this is an extreme case here, for instance, where when you take out all of the other non-canonical ranks, you end up with this genome being classified just at the phylum level. And we always classify all the way down to species. There are link outs to the NCBI pages and other information that comes as part of the QC, but that's basically where we draw the line. We have produced a software toolkit called GTB-TK so that people can assign their own genomes to the GTB taxonomy, and that's been getting pretty good uptake. So how are we doing? So thumbs up, it's getting used. We're very happy to see that. So we've had more than 40,000 unique visitors to the GTDB webpage since its launch, and very nicely, nearly 2/3 of those visitors will come back for multiple occasions. So that means they're actually using the resource. The primary publications that of the resource, which is the initial publication in 2018, the toolkit publication in 2020, which is Charme, and then the ANI-based species clustering in 2020 have collectively been cited over 1,500 times. So that's telling us that people are using it and publishing with it. And then we also have adoption of the taxonomy by third parties, including Bergey's manual. So Bergey's manual have been using Gtdb. When people are contributing chapters, they're encouraged to have a look at what Gtdb is doing as another taxonomic resource. Silver, as I mentioned, uses it. So we interface with those guys at the 16S database, and Anvio also has plugins that use the Gtdb taxonomy. Down the not so great feedback, there was a paper published at the end of last year called Microbial Taxonomy Run Amok. And this is basically pointing its finger at Gtdb as the main thing that's causing the run amok. So we have introduced quite a number of changes, and a lot of them, like a lot of the people that are very used to a particular taxonomy, don't feel comfortable with that. And there are quite a number of very valid concerns shown in this, written into this paper, which I recommend everybody to read. But the bottom line is that they say that what we're doing is creating a Wild West situation, which is ironic because we're trying to do the exact opposite, trying to avoid a Wild West situation. But some people believe that we're actually creating a Wild West situation. So the valid concerns in this paper are really what feed into the lessons learned that we've had. So I'll walk into that now. So first of all, when we started, we walked in fairly nomenclaturally naive. In fact, I did not even make a distinction between nomenclature, which is the naming process, from the taxonomy, which is what we would define as making the framework upon which the names sit. So I had not really thought it through. I was aware that there was a prokaryotic code. I hadn't read it. I didn't know the rules particularly. So we just sort of walked in blindly and started putting in names, which turned out to be a problem. So the prokaryotic code, or ICNP is the acronym, they have rules such as higher ranks must be based on a genus stem, which was introduced in 2008. We're sort of aware of that in that you see the names and you go, oh yeah, the genus Burkholderia is within the family Burkholderiaceae. So get the idea that you use the stem and then you add a standardized suffix. So in the case of family, it's Aceae. But I didn't know necessarily the rules. I just knew that I was just seeing it and then sort of working it out without actually consulting the code. So something like the beta Proteobacteriales is a big no-no because that is not based on a root, a genus root. So the name itself was not valid under that rule from 2008. The gamma Proteobacteria is okay. It's actually validly published, but technically after this rule was implemented in 2008, it's not technically correct. And then the rank of phylum, as I mentioned before, is not formally recognized under the code. So basically anything goes. And so that's, names like Proteobacteria and what have you. They're not connected at all to any genus name. Now, just recently, the ICSP, which is the governing body of the code, have voted bringing the phylum into the code. So that will be interesting to see how these kind of legacy names are going to be handled. I imagine there'll be a full-on riot if people change the name of Proteobacteria or Firmicutes. So this we changed in the later one to Burkholderiales so that at least the order was following the expected trend. Okay, so what we did do, I mentioned before that it just seemed very logical that we would go in and create these new higher rank names like this, but we didn't make any formal proposals, and that's been a real issue that's kind of bitten us in the arse a bit there. So here we see what's happened is by making these names en masse, and we're talking hundreds of new names that we did in the initial release, they're not published. So that and when you propose a new name and you publish it, that's an effective publication called an effective publication. And then after it's picked up by the governing body and put on a validation list, then it's validly published. That's in fact the only code that does that. But essentially you need it to be at least effectively published to move ahead. And so this was a paper describing a new genus and order, and they said although present in the GTDB, formal proposals have not been made and the names have not been validly published or effectively published, and therefore they have no standing in nomenclature. So this has turned out to be a problem. But what these guys did was that they actually used the name that we proposed or didn't propose, or that was in the taxonomy. So that's now proposed in this publication. But they could have proposed something else, which would have to overwrite what we did because we didn't make an effective publication. So we've now, after we recognize that error, we flag them now within the taxonomy. So if you go to the browser in GTDB, you will see names, and if they have a hash behind them, those are Latin names that we came up with that we were too enthusiastic. So this is lesson 1. We were too enthusiastic with Latin names in the beginning, right? And so here's an example. We knew that this group Thermodyselpha vibrio was in its own family, order, and class. So we simply added the standardized suffix ACA, ALES, and IA is the class suffix, but we didn't propose it. But that was just logical, right? But it's going against the way things are done in the code. So there are actually now 435 of these not effectively published Latin names that you'll find in GTB, but you can find them through the hash. What we are seeing is that here's a paper published in April just very recently that as part of their description of a new genus, they go ahead and actually effectively publish those names. So now with that, we're down to 432 GTB Latin names, because this would be great if it happened and people just started using the names, but it's not always going to work like that. So the current plan is that we'll make one large publication putting these names forward and effectively publish them that way. However, I do want to point out, like, we've had quite a few people ask about the phylum names. So people are not used to seeing these names with OTA on the end of them, but that's not actually us. That has actually been proposed. So the new phylum names are effectively published. That was in 2018 by the leaders in the nomenclatural field. And the proposal was to use this because at the moment phylum can have any ending, and so the proposal to use a fixed ending of OTA, and that was put forward, and that's actually been put to the vote in the ICSP and has been approved now. So these are actually the new names using the standardized suffix, and here are the existing names. So that's not us. In case you want to say, why are you doing that? We've had quite some people saying, what are you using these OTA names for? It is actually in the literature. And I think makes sense as much as possible. Why wouldn't you have a phylum name standardized like you have the other ranks standardized? So our current practice now, as we continue to curate, is we don't use Latin names unless they are at least effectively published. So here's an example where we find in the tree, Cyclonatronum is in its own family under what we call the order Balneolales. So in the past, we would have just turned that into a family name by putting the standardized suffix, but now we just use a placeholder name. So we don't confuse the system more than we already have. But that's then open. Somebody could go and propose that if they agree with with our taxonomic structure. Okay, now I mentioned type strains all along. Type, species, and strains play a really critical role in nomenclature, and you need to look no further than the Cyanobacteria to understand this. So Cyanobacteria are unique in that they've been under the botanical code for so long and barely under the bacteriological code or prokaryotic code. And so their classification comes from a rich background of phenotypic classification. Here's a picture of a genus Toleopthrix, and it's got beautiful large cells, and that's what the classification is based on. But when you put that into a tree, that phenotype is not monophyletic. So here in the tree, you see Toleopthrix turning up in several different lineages, and you need a type species and types and a genome of types species, more to the point, to anchor the genus so that you know which is the true Toleopthrix, right? And that's a good illustration of why type species and type material is so important within nomenclature, because they provide the anchors or the flags, the flags that tell you where you are in the tree and what the name should be. So the lesson 2 then is beware of taxa without types. Now, what's happened here is that there have been some nomenclatural loopholes which have allowed the proliferation of Latin names without type material. And that comes down to all of these uncultured taxa that are now turning up, courtesy of sequencing directly from the environment, or so we don't have a culture of it. And so they have been accommodated since the mid-90s under appendix within the code for candidatus, but there's no rules really around the use of candidatus. It's very open, and it's not part of the code. It's outside of the code. It's not recognized. It has no priority. So these candidatus species have been, adopted to a fairly large extent by particularly microbial ecologists. And so it's just been anything goes. You can just plug a name. It doesn't have to be connected to type material. And so here's a good example of where all of these new candidatus phyla have been proposed as part of the characterization of the candidate phyla radiation. So none of these are actually connected to any type material. And that's a problem for the same reason I just showed you with the cyanobacteria, because if any of these groups break up in subsequent trees, we don't actually know which group the name should be associated with. That's the problem. That's also compounded by the fact that phylum is not an official part of the code. So even if it was an isolate, you could still do anything at the rank of phylum, where a lot of the activity has been going on. So this really stems that uncultured taxa cannot be named under the code anyway, because you require pure cultures upon which to base your name. And so all of these uncultured taxa are basically outside of the code, with the exception of that tenuous script via candidatus. So that's a problem. So we wrote a paper in 2019 and that proposed that we should be using, actually we weren't the people that initially proposed this. This was already proposed in the literature by Barney Whitman and others. But we said, if you're going to give names to uncultured taxa, please, please, please use the genome's designated genome as type material. So we've got an anchor and we know where the name should follow. So we wrote a little paper, the importance of designating type material for uncultured taxa. And so this was a call out because, I named the bacterial phylum once. What do you call it? Bob. And effectively you can call it candidatus Bob under the current rules. And then you can give it a name, but we want it to be consistent. We want it to be connected to a specific genome. So there's a type, there's nomenclatural type upon which that name can ultimately be based. And I just want to flag also that there's now a new code, a proposal for a new code called the SEEK code, or its working name before that was Uncode. So you'll sometimes see it referred to as Uncode. And I point you towards this website at BISMiS. There's the link. Which has a lot of really good information about this initiative. But the idea is to create a new code around genome sequences rather than axenic cultures, which will free up, that means that we'll now be able to put in all of those uncultured taxa and get them within a code. So I point you to that. So the third lesson is that ultimately it's often still a matter of taxonomic opinion. So this is the old splitting versus lumping argument. So the curators, I'm one of the curators at Gtdb, will often have these arguments about which way should we go. So Gupta et al. Proposed a few years back that the genus Microbacterium should be split into 5 separate genera, and that's reflected in release 89. Mycobacteroides, Mycolibacterium, Mycolobacillus, Mycolibacter. And then just recently, it's a preprint, there was a proposal based on reconstituting it into a single genus again, Mycobacterium, which was reflected in release 95. The bottom line is that actually both of these solutions are viable under Gtdb. They are circumscribing either 1 or 5 monophyletic groups, with the exception of this one here, rogue one. And they are within the range that is acceptable in terms of relative evolutionary divergence for genera. So we could accommodate either of these solutions, and we have. So what we end up doing is just follow the prevailing taxonomic opinion. And that's as old as taxonomy, that there's taxonomic opinion and there'll be people with different taxonomic opinions. Now, the last lesson, and this was one of the ones that was really called out in the Run Amok paper, was that we should engage the community more. So we actually did try, in that paper, they actually singled out our recent reclassification of the Delta Proteobacteria into 4 phyla. And we did try to engage the community by picking out people in the research community that are, heavily interested in the Delta Proteobacteria and that would be interested in restructuring. I think it's fair to say that people have recognized for a long time that the Delta Proteobacteria are not Proteobacteria and they're not monophyletic. So that wasn't a surprise, but nobody had actually gone to the bother of reclassifying it. And that's what we did. But the Run Amok paper said that, we basically were biased towards people that are used to using sequence data for taxonomy, which is very true, and that we didn't really call upon people that know the Delta Proteobacteria in a very deep physiological way. So that is a reasonable criticism. So what we've done is we have initiated a forum, which is getting a little bit of traction. And this is where people can post and say, what the hell are you guys doing reclassifying the Delta Proteobacteria? It makes no sense at all. You should be doing it this way. So this is an opportunity for not only for the user community to talk to us, the developers and the curators, but also to other members of the community. And what would be lovely is if it gets debated on there, and then we'll take the consensus of people's comments. But the bottom line actually is that most people visit the Gtdb, they just want to be told what's the taxonomy. They don't necessarily want to get engaged in a debate about what it should be. They just want a consistent taxonomy that they can use for their research. So I'll just finish then. Oh, good, I haven't gone too long. All this aside, all of the teething problems and the lessons learned, we should be very, very cognizant of the fact that we have a long way to go. So a recent estimate, and this is based on 16S, so it's probably even an underestimate, is there's an estimation that there may be 2 to 4 million prokaryotic species on the planet. And this is the different releases of the Gtdb here. And this, keep in mind, this is a log scale. So these are the number of phyla, classes, orders, families, genera, and species that have been identified or named between the releases, which includes uncultured taxa. And so the current number in R202 will be somewhere in the ballpark of 45 to 50,000 species. So that means, based on this, we've still got 99% of species yet to be described. So just keep that in mind. And in order to do that, to go that long, we really do need a systematic, scalable taxonomy. It's going to be essential, whether it's Gtdb or something else. We do need it, because if we don't do anything, it's just going to get more and more chaotic as we fill in the gaps. And I mean, the really exciting thing, of course, is that we are going to be able to fill in this gap. We're getting, as we saw earlier on in my talk, we're getting so many hundreds of thousands of mags now. We are going to circumscribe, hundreds of thousands to millions of species in the foreseeable future. And if you think of it in terms of a database problem, actually a database that's got 2 million data points in it, it's a pretty small database. It's not that big from the data angle. It's just that we need to have a robust, systematic way of putting that diversity into the framework. All right, so I'll finish here. Just acknowledge the team. This looks like I'm acknowledging myself. I wasn't going to put my name on myself, but I just felt it was weird not to. So I'm really acknowledging the team here. Great team. So Donovan, Pierre, Adam, who's left us now and been replaced by Aaron, there are the developers. And then Marsha, Chris, and myself, we're the main curators on the database. And it was funded by, what happened there? And it was funded by the Australian Research Council, so very happy to have that funding. Thanks for your attention. Happy to take questions.
Thank you, Phil. Thank you for that wonderful talk. Excellent to know what all we've been going through for the past couple of years and how we have progressed in this field. Phil, one of the first questions is, this has been asked by Preben Nielsen. Does it make sense to take years when you talk about evolutionary distance? Wouldn't generations make more sense?
Well, we're not really at this point in where we can calculate generations from the sequence data. We can calculate an evolutionary divergence. We calculate that as part of the inference, but generations, well, we know what E. coli can grow in a rich broth. It can replicate in 20 minutes, but if I stick it into water, it may replicate every 2-3 days or not at all for a while. So I don't think it would make sense to express it in terms of generations. I think that's very dependent on where the organism is present. I get where you're coming from though, because what you're saying is that the evolutionary rate will depend on the situation of the organism, and generation time is one of the metrics that would tell you that. But I think it'll still work because if you have the tree and you know how far that's evolved and you can calculate the timing point, the relative timing point, then I think it's still valid that you can surmise that they coexist at the same time. And actually there are data now coming out. There was a study by Antonis Rokas' group applying this RED approach to fungi, for which they have some dating information. And so one group that they calculated as being much younger than was suggested when you corrected for that evolutionary distance is consistent with the well, as far as you can tell, looking at fungal morphotypes under the microscope, it seems to make sense. Okay. Yeah, interesting question.
Yeah. And then Amit would like to take the next question. Amit?
Yeah. So next question from the Q&A is again from the Preben. They're using Gtdb extensively. And then the person says, I find sequence-based frameworks for the taxonomy is very useful, but the taxonomy is for the biologist. Therefore, the naming bacteria requires physically available or deposited organisms. We are many microbiologists that are dependent on the physical availability of the strains via culture collections. Can you see a situation where the framework is used, but the formal description requires scientists to document the physical existence of organism?
So you don't think that a genome sequence tells you that the organism physically exists?
That's the question by Preben again. So
yeah, no, I mean, if Preben's around, maybe I can answer that. But I mean, I think the fact that you've got a genome sequence tells you that the organism exists or has existed.
Yeah, if the Preben is there, she can unmute herself or himself and can ask or can answer.
And I'd also say you can tell an awful lot about an organism from looking at its genome sequence. You can do a metabolic reconstruction and you can work out what it should be capable of doing. So I would say many microbial ecologists would argue that point, that you need a deposited. It's certainly, nobody would contest that it really helps to have a pure culture to do experiments on. And there's if you go to that workshop, the seed code workshop, there's a really nice talk from Kostas Konstantinos where they identified the organism, the dominant organism in an oil spill, and they pulled back the mag of that organism, the whole gene, and then they worked out from what it predicted to do, how they could isolate it, so-called genome-directed isolation, and then they got a pure culture. And they got the same sequence back and they got the isolate. So in that case, I'd argue it's actually better to go in via sequence-based methods because it's pointing to you towards the most relevant organism in the ecosystem rather than just what normally grows on a plate, which would be a weed, which may have nothing to do with the major function of the ecosystem. So I say these are not competing things, these are complementary things. And unfortunately, it's often put down as a competition. You either do cultures and isolates or you do culture independent stuff, but they're complementary methods.
So, related question to that is, and in fact, you answered partially what qualities, if the phenotype is important in the microbial taxonomy. Maybe Phil, if you can just summarize very shortly about the seed code, maybe all these questions are related to that, if I'm not wrong.
So the seed code is, there was a question that was put on to the prokaryotic code, the ICSP, asking if we can start to use genome sequences as type material. So I think there's a lot of compelling reasons to use genome sequences as type material because they're easy to store, electronically. Anybody can get hold of them. If you're using an isolate, you need to get it from a culture collection. You need to be able to grow it off, they may be contaminated, other things. Whereas you can download a genome sequence fairly easily. The main functionality of a type material is that it unambiguously identifies the organism. So certainly a culture does that, but actually the requirement for a culture is relatively new. Did you know that even 30 years ago, it was acceptable to submit a drawing of a microorganism to use as type material, which is in retrospect fairly, I think, dangerous. And there are still a few legacy things like planktomyces that are based on visual descriptions of the organism. So I would say, a genome sequence is, you're getting the blueprints. You have the blueprints. It's the most unambiguous way you can identify an organism. and you can use that in order to get a culture and then do some experiments on it as well. So the seq code, we tried to, so I sit on the ICSP, and about half the people on the ICSP were looking to put a proposal up to use genome sequence as bona fide a type material, which would open up all the uncultured taxa. Now you could describe an uncultured taxon using the full weight of the rules in the prokaryotic code. They would have to be bound by the requirement for type material, for naming conventions, all the rest of it. And importantly, they'd have priority. So if somebody else comes along after you, identifies the same organism, they have to use your name because you've got priority. At the moment, there's no requirement for that for, say, Candidatus species. Somebody can come along and get an isolate. It's identical to a Candidatus name that's been proposed. They can propose a completely separate name. So something had to be done. It was rejected by a 2/3 majority. It's just too radical for traditional taxonomists. And so we've gone to a plan B. There's a good paper there's a paper there, a consensus statement about what this is all about. And the plan B is to set up a parallel code which does use genomes as type material, so people can propose names based on genome sequences. It accepts all of the names, valid names from the prokaryotic code, and so it's trying to streamline as much as possible with the existing code. But that's with the major goal of opening up priority in genome and genome-based taxonomy, so that we can really start to name the microbial majority. I see also there's a question in here, only 18% of taxa have valid names. How long will it take to overcome these challenges? Well, actually coming up with the Latin names, there was a paper recently for a resource, the acronym GAN, and that was proposed by Aaron Oren and Mark Pellin. And they showed through combinatorics, you can actually generate millions of grammatically correct Latin names for organisms. So I don't actually think that's going to be a bottleneck. I just think making sure it's coordinated, that things fit into a common taxonomic framework so people aren't naming the same thing with different names, and that I think it's actually doable. I mean, if it really does turn out that there's 2 to 4 million species on the planet, I think it's eminently doable. Just in a more ecological point of view, I think what's really going to happen is that you'll have this rare biosphere. It's going to be harder to get those taxa. So maybe there's 2 to 4 million more common species on the planet. Is that kind of answering the question?
Yeah. Phil, I would like you to answer Ian's question, which is stability in nomenclature is highly important. So what happens if Gtdb classifications change with each release? Is this going to be problematic?
Well, I would say that nomenclature faces these problems anyway. I mean, there are people with different taxonomic opinions, and they make suggestions for changes. So I think that's just part and parcel of the exercise. I would hope that, in fact, it would have the reverse effect. If gets enough buy-in, people like the phylogenetic framework and are good with it, that should actually help stabilize things, I would think. What we're seeing, we know that we've done comparisons between releases, and in terms of the phylogenetic framework, the named nodes within the tree, about 99%, 98.6% of nodes are very stable between releases, even with adding in a whole bunch of new leaves into the tree. There are a few problem areas that tend to jump around, but for instance, that Microbacterium example, that's not down to the tree, that's stable. That's down to the taxonomic opinion of individual researchers, right? That's not Gtdb's problem. They're just trying, you've got more interior nodes that you can put names on, and stable interior nodes than you use, because you're only dividing it up into 7 ranks. So you can put, as you saw in that example, you can put additional names on.
Yeah.
Does that answer the question?
Yeah, here's one question from Feng Ping. Since Gtdb is now endorsed by several organizations, does it mean that it is now obligate to use Gtdb to make valid taxonomic descriptions?
No.
At least based on uncultured.
No. Okay. No, it's taxonomic opinion, so the nomenclaturists are very strong on freedom of taxonomic opinion.
Right.
So we can't come along and say you must use this taxonomy. We just can't do it.
Yeah.
But we can certainly flag things, like we see quite commonly where people have given 2 genus names to things that are really closely related to each other. That's usually because they're naming them very close to each other, and so we can help to in coordinating the traffic so there's no traffic jam. So
Yeah.
and we can propose, we provide tools that allow people to use it. I think what happens is a lot of people, they're not so interested in the classification part of it. They know that they need to do it, so they're very happy if there are tools available which help them do it, so they don't have to spend a lot of time, doing it themselves. So we assist, but we certainly don't dictate.
Okay. Here's a question that has come probably from one of the younger students. What qualities one should possess to become microbial taxonomist?
It really helps if you have obsessive compulsive disorder. So you've got to really like, trolling through thousands of names and cross-checking databases and stuff like that. All our curators have that problem. Or that you're good with the idea of banging your head against the wall repeatedly. That probably helps too. Sorry, I'm just joking, but I should say, I think it's a very exciting area, and I think it's a very basic area that you need in order to, it's a foundation upon which a lot of interesting science is built. It does have a bad reputation, but I think it's essential.
Yeah. So I would like to just add to this question here for the young microbiologists or who are interested in microbial taxonomy. One of the biggest limitations is the knowledge about Latin language. And that's been one of the sort of bottlenecks here. And if you are really interested in learning Latin and try to name organisms, you're more than welcome to join us and become a trained nomenclature. So that's one of
actually there's surprisingly not too much formal training in the area.
Right. That's the biggest limitation, actually.
Yeah, most people pick it up by osmosis.
Yeah.
Which is not the best.
So here's another question. Microbial classification in current practice is basically based on both the phenotype and phylogenetic data. But Gtdb is generally based on the evolutionary data from genome.
Yeah.
So how valid will it be to use Gtdb for classification, since majority of the depicted genes in the genomes may not be expressed in the environment?
Yeah, fair enough. So that's referring to polyphasic taxonomy. So my answer to that is that if you're going to do polyphasic taxonomy for 2 million bacteria, good luck to you. You have to have something that's scalable. But more importantly, I feel, and this is what I believe, this is my opinion, that the genomic framework is telling you what the species trajectory is. So that's the trajectory of the species. Now we know that genes travel left and right, phenotypes change. Some are like plugins, you pick up different genes from the environment or neighbors. And I think you use this framework in order to address all the other stuff. So I think what you actually do is you overlay all your other polyphasic information onto that, and that helps you to interpret all the other information. But I don't think, and I strongly believe, it shouldn't be part of making the framework. I think that you make the framework from the core vertically inherited set that identifies the species evolution, and then you overlay everything else, and it's very effective. You can really understand what's happened ecologically and evolutionary using that approach.
Right.
So I'm not a big fan of polyphasic taxonomy from that point of view.
I would like to add my own question here. So how have you seen the genome relatedness indices change over a period as you keep adding new taxa or new sequences into the Gtdb? Because I remember when ANI and all these things were first released, there was a single threshold value, but now there is a range within which ANI should be used. So how do you think this particular matrix is going to change over a period as you keep adding that 100,000 genomes?
It seems to scale pretty well. Using ANI to define species, which is really an operational definition, and is a necessity currently for the scaling of the problem. But it actually goes against the basic principle of using, say, relative evolutionary divergence to define the ranks. So it's using a threshold. It's using a fixed threshold. But because you're close to the tips, I think it's by and large an acceptable operational definition. So if you want to read more about how we actually applied ANI, because we certainly aren't the only people that use ANI to define species, it's very common, and it was originally suggested by Kostas Konstantinidis.
Right.
But we center those groups around type material. And then anything that doesn't fall within the radius. So we have a number of rules, just operational rules, that if you have two type species that are closer than 95% but less than 97, then we define a smaller radius. It's still two separate species. If you have them higher than 97, then one becomes a synonym of the other. They can't both exist because they belong to the same species. But you know what my dream is, or my vision is? I would love to see a biological species concept supersede that. And so there's a nice paper from Howard Offman, a couple of papers where he's using recombination boundaries as real biological species. So just the same way that we define animals by recombination boundaries, that between species we can't reproduce and produce viable offspring because there's a recombination boundary, and but there's no recombination boundary within a species. He's shown computationally you can take genomes and estimate the same thing. And that would be, it's very computationally intensive at the moment, but if that holds up, that'd be a great way to define the base level of the species, which is, you know, arguably the most useful classification if it's actually based on a biological species rather than an operational construct. And keep in mind, the whole thing we're doing is an operational construct because Tree of Life doesn't get divided into 7 ranks. We divide it into 7 ranks so that we can communicate about it. But that's not necessarily how evolution's worked it. So yeah, I'd say I'd be very happy to use a real biological species concept if that method will scale.
Okay. Here's another question from Huibin Lu. Whether there is a reasonable boundary to distinguish different genus, family, order, or higher rank based on the 200,000 mags in Gtdb. Have you been developing any indicators? I found some propose 70% AAI as the boundary for determining the genus.
Yeah, so using AAI is basically just using a sequence threshold again. So it doesn't take into account different evolutionary rates. So you basically bake at the same mycoplasma type of thing. So 70% AAI is just a fixed threshold without taking sequence divisions, relative sequence division into account. So that's the whole basis of using the red, that it's contouring, and that's what we're using. And they're fairly generous ranges, and they slightly overlap, but it's ordering them into, wrangling them into these more or less discrete channels. And so that it scales, and we don't use AAI for that purpose.
Okay.
For that reason, because it doesn't take into account, and you, of course, because it's a human construct, you can argue, well, I don't care, I want to use AAIs as my boundaries, and you can use AAIs as the boundaries. There's nothing to stop you, and some people do. So, but I would argue that my preference is to take relative evolutionary divisions into account for the reasons I gave.
All right. Another question from Wheelie Lin. As more new species are added into the phylogenetic tree, some clades may often be changed and not be stable when the Gtdb is updated. So do you have any evaluation methods to provide confident values for some specific clades which indicate its stability in the phylogenetic tree?
Yeah, that's a good question. We have spent a fair bit of time testing the stability of clades, and it is, to my mind, remarkably stable. There are definitely places which have poorer stability, but for example, the average bootstrap support for the named nodes that we use for the taxonomy at 97% is the average. But there are some that we're obliged to sometimes use some poorly supported nodes. We know it's going to be unstable, but we don't have necessarily a good other option for that. Okay. We do provide the tree with the bootstrap values. You can download it from the website so you can see that yourself.
Okay.
But I would say it's been pretty robust between releases. We also have a tool which is a taxon history tool, so you can see what the name is in previous releases, and you will see changes. And often these changes are not actually a result of underlying weakness in the tree. It's like the Microbacterium case. It's a taxonomic opinion. Right. So there's a new study. It's now 5 genera. It's now 1 genus. So you'll see changes that are down to changes in ongoing taxonomic opinion.
Yeah. So regarding that Mycobacterium, Ian had a comment. He basically is answering your question. I asked about Mycobacterium because it looks from your slide, you were saying that the new release collapses 5 genera back into 1.
Correct.
Is
Well,
that
that was
right?
already in 95 because we were following the LPSN. So they have a taxonomic opinion. We follow that as well. And they said they have a thing there where they'll give the name and they'll say this is a synonym of Microbacterium, and that our taxonomic opinion is that it should be Microbacterium and not the other name. So we follow that. I mean, really, in essence, what we provide is the framework. And as much as possible, we follow what the community is telling us for the names. Right? So that's how we view it.
All right. We'll take the last few questions. So one is from G.H. Zhou. You use a heuristic alignment algorithm. The alignment will be affected by the sequences. The problem is that when you update the database, there may be some change in the alignment sequence as well as the taxonomy. So when there is some difference in taxonomy for one sequence, which one we should believe?
Certainly, as you add more sequences to the database, your align. We do use a fixed alignment from the markers, but we've also played with subsampling the alignment, and so we have spent a lot of time assessing how stable is the phylogeny with using different alignments, different masks, different inference methods, different models. And more or less, it's stable. You definitely will have some taxa which will be unstable and will change between releases. So that's basically the answer. So which one do you believe? I mean, okay, imagine Gtdb's there. You'll have different taxonomic opinions. Which one do you believe? You research it. You find out which one. If you're working on a particular group, it's just another source of information that you can refer to.
Okay. Last two questions. So Jun Liu is asking, every 6 to 8 months, you will update the database or may update the taxonomy. How can we adopt to this change?
How can you adopt change? So what I would say, if you use the taxonomy, refer to the specific release. That will help. And you'll see, I mean, if you go in and just poke around, some taxa are completely stable. It's exactly the same every single release, no change. So that's what people have been doing. They have been referring to the Gtdb taxonomy. If they're naming a new organism, I would always recommend that people make their own trees and have a look at it and evaluate the data in different ways. So again, it's an assistant. It's a tool to help with that process.
Okay. I think it's one of the last questions. Does Gtdb database allow extraction of single copy orthologs?
So, yes. I mean, there are alignments, and you can also, I think, get the individual markers separate. I can follow up on that. I think we've had requests for that before. So there's a repository of information. Oh, by the way, I just noticed one in the chat. There wasn't a question that somebody said. Alessandro said that Tolepothrix does have a type. It's true, Tolepothrix distorted, but it's not sequenced. So there are named types, but they haven't been sequenced. And so we actually are part of a international consortium that are making a concerted effort to identify type cyanobacterial species and then sequence them. So to provide all those missing flags, all those missing anchors.
All right. I think we've had all the questions. We've covered most of them. There may be one or two that are left.
Thanks everybody for all the questions. There's a very active crowd.
Yeah.
Very impressive level of interest. I'm sorry if I didn't get to everybody's question.
No.
I think the talk will be available, right?
Yeah, the talk is available on YouTube. So as soon as this live session ends, we'll edit it and post it back. So it will be available. Yeah. Thank you, Phil. Thank you for the wonderful talk and discussing with our participants about how the Gtdb is helping in changing the course of microbial nomenclature and systematics.
You're most welcome. It was very enjoyable, and thanks for the invitation, and all the best for the series. I will try and tune in.
Yes.
I think Ian Sutcliffe's talking next month, so that'll be well worth tuning in for.
Yeah. Thank you so much.
All right. Thanks, everybody.
Thanks a lot.
See you.
So
There is a question from one of the participants, and maybe Kamlesh can answer, is that how to become a BISMiS member?
oh, okay.
Maybe that is a question that you can answer for everyone.
Yes. BISMiS has its own website. It's bismis.net, bismis.net. And we have special memberships for students and some special memberships for faculty who would like to just be a member and be part of all the events that we conduct. There are few additional perks. So the membership rates are decided based upon what you would like to have. As I said in my introduction, the membership fees is divided into two parts. One is with the Burgess Manual for Systematics of Archaea and Bacteria. So you have a complimentary online access, and it is provided by Wiley. So the charges for that are for a full member, it's about $50. And for a full member without the BMSAB access, it is $30. And for a student member, the access to BMSAB is at $30. And for for membership without the access to BMSAB is $10. And you can become a member by going through our website. There is an online portal by which you can pay the fees. Even though you do not have the access to BMSAB, there are a lot of additional perks. You still get the voting rights. You get priority access to the meetings. You get priority access to these Burgess or BISMiS talks. The series, you get voting rights. You can contribute your articles to the journals. That is the Bulletin of BISMiS, and also to the Microbial Taxonomist, which is published by the Burgess Manual Trust. You can be part of the BISMiS and still have access to all of these voting rights. You can be part of the discussions that happen in-house. You get discounted registration. So a lot of these perks come with it. Visit our website at bismis.net. We have not been so active initially, but now we are trying to be more conclusive and involve more participants from all over the world. And now you'll be seeing more of us. Subscribe to our YouTube channel, subscribe to our Facebook page, subscribe to our Twitter cast, so you can learn more about BISMiS and how we are helping. And thank you so much. And if there are no more questions, I think we'll close this inaugural session. Thank you to all the participants. We had about 200 participants logged in through Zoom, and about a 50 odd who are watching through live YouTube. We thank you all, and please fill in the survey link, because we would like to know the times that are more convenient for you. We'll try to incorporate them as soon as possible, and we'll like to have more involvement from everybody.